Okay, well, that was almost a disaster, but yeah, as you guys have heard, I'm a bit of a substitute, so not quite fitting, and hopefully not a total disappointment despite a small delay. But for those of you who may be just a little bit disappointed, let me start by placating you and saying, okay, yeah, it is working. And saying that this conference really shows how much has happened in the past few years to, how much we have learned thanks to, including all the people who are in the audience, how much we've learned about how the chromatin behaves, what's its function, how, more broadly the genome folds. But I do want to point out that we've learned a lot about how the human genome folds. And in order to illustrate, I'm actually showing you here this classical slide from Erez that he showed you yesterday that keeps kind of, migrating from one presentation to another presentation. I'm just going to click because this thing doesn't seem to be doing what I want it to do. To highlight, that there's this key word, the human genome, how the human genome folds in 3D. And if you just remove that crucial word, the human, from that slide, it kind of doesn't work anymore because a lot of the methods that you've heard about in this the bunch of microscopy methods that have been presented and a bunch of contact mapping methods that we in the lab especially use a lot, they stop working unless you know the sequence of the genome that you're interested in, the one that folds. And so, so in order for you to study anything of the kind, in order to use all the methods that have been developed by the people in this audience, in species that are not human, you need to have the human references. Oh, sorry, the reference is kind of akin to the one that we have for the human. So you would say, what's the problem? Well, the problem is that the genomes are kind of long. Here's a slide that kind of gives you an idea of the scale of what do I mean when I say long. And so what does it mean that they're long? That usually means that reading the genomes is also kind of long and expensive, right? So famously, reading off our own genome has taken 13 years, $4 billion, 6 countries, 20 research centers, and basically, that has ushered, major advances in the human studies, but kind of made relatively little for the 8.7 million species, that are non-human on planet Earth, or whatever the latest estimate for that number is. You at this point may be confused, right? Like, haven't you heard about Illumina saying that now you can have the genome for $1,000, and a few weeks ago, maybe for $200. You would keep track of that. You'd think that they pay you for sequencing the genomes, not the other way around. So just to kind of clarify a little bit, what do people mean when they say, you can have that genome for 1,000 or 200 or whatever it is right now? What they mean is that for that number, you can have the collection of short bits of the genome that if you extract the genome from the cell, you chop it up into short pieces, you read it off, you get this cloud of different reads. But in order for you to make any sense of them, you still need to align them to that 4 billion reference, right? And as you kind of align, you start accumulating, you notice something interesting that is for the particular, let's say, individual in the clinic. And so what you get for that relatively little amount of money, you get a collection of these bits that are different in this particular individual, and but again, with respect to that $4 billion reference. For everything else, you kind of have to solve the problem kind of from scratch. So what do you do if there is no reference for you to align the reads? You start aligning reads, to each other. And as you do that, you notice there are some overlaps. You merge them together to get some contiguous sequences, aka contigs. And this works great, except for it doesn't work all the way. The problem is that the genomes contain repeats, and as you hit a repeat, the overlaps lead to ambiguities. And so instead of, let's say 46 very long sequences for something like a human genome, we get 100,000 of very short contigs. And, nobody likes that. It's very difficult to interpret. You get the end of the gene here. You don't know any sort of control sequences anywhere. Basically, people really want to have something that looks more like chromosomes. So what do? They bring some sort of external data that will tell you that this contig and this contig, they kind of live together. Maybe you need to reorder them in some form. That leaves gaps in between, but you get something that kind of contextually is a little bit like the chromosome. This is called scaffolding, and this is kind of where much of the money when assembling genome, went into just up to a few years ago. The reason for that is because different repeats meant that you had to kind of jump across different length scales when you tried to scaffold. Different methods were better at different scales. You had to bring all sorts of specialists from different fields in order to do that, and that costs, of course, a lot of money. And even in those cases, very often you didn't get whole chromosomes. So kind of a few years ago, there has kind of emerged some excitement about using the Hi-C data as a powerful source of linking data. So in order to get you an idea of why people thought that this data type might be interesting and might be useful, for the purpose, let me kind of recap to make sure that everyone's on the same page, although we've seen a version of the slide a few times already on this conference. I'm not going to take you through the details of the reaction. I'm just going to tell that, right, what we're doing in this Hi-C is that we're translating the information that is kind of hard to read in high throughput, the proximity in 3D, we're turning it into the information that we're really good at reading at high throughput, which is the proximity in 1D. So, right, we create these chimeric sequences and we create billions of them in one experiment, and then what we go ahead and we do is we map the bits of those sequences to the genome of interest, and right, like every single read pair, every single chimeric read yields you a point on the map. And right, like usually we represent this billions of chimeric sequences as a heat map, which shows, like here on the scale from white to red, the bits of the genome that talk a lot, which are very red, to the bits of the genome that talk relatively little in 3D, which are kind of white on this contact map. And so if you look at it for a bit, you'll see even from this zoomed out human genome map how this kind of data can be useful for genome assembly, right? Imagine you have 3 contexts, and 2 of them talk a lot to each other in 3D, and they don't talk at all to the third one. So, right, that probably, if you look at this, you can probably guess that the first 2 you should probably anchor to the same chromosome, and the third one should go somewhere else. And if you zoom in, right, there'll be a lot of interesting signal that we're all really good at interpreting at this point, but the first order of signal that is there is this thick diagonal that is slowly decaying away from the diagonal. Broadly speaking, anything that is kind of closer together in 1D will have more contacts than anything that is further away, which means that you can use the frequency of contacts as some measure of distance, of 1D distance within the chromosome, which means that you can use it to order the contigs and reorient them as well, because orientation is just the distance to, one end or the other. And so, when people realize that this signal is useful pretty much, from 2009 when the first version of Hi-C, the high throughput, 3D chromosome conformation capture method has come out. There's been a bunch of methods, really great work where people would try to do proof of concept, reassemble existing genomes, add Hi-C as part of this, other types of methods that people use for scaffolding, but overall, pretty much as of say 2016, there was still no cost-effective strategy to assemble genomes end-to-end. So much so that when we found ourselves in the previous healthcare crisis, the Zika epidemic, the New York Times actually came up with this article on the front page, right? It's the assembly problem that they presented, and the assembly problem was for the yellow fever mosquito. It's the primary vector for the Zika virus. And so as far as I know, at least as of right now, the only time when the New York Times has put an assembly paper on the first page. All those bits and pieces, those are individual contexts that people didn't know how to put together. And so at the time, we realized that we've had a bunch of methods that seem to have worked in order to solve this problem, and I'm just going to give you in the next few slides an idea of how they work. Don't worry, I'm not going to take you through algorithms. I'm just going to, my goal is to try to give you an intuitive idea of how they work, and I believe that it's possible. At least my kids seem to be able to understand the whole thing intuitively enough, so I hope that we have a good chance for success. Okay, so imagine that you have, assembled your genome somehow, you've got 3 contexts, you don't know how they belong together, you have no information that tells you how to properly put them together, whether there may be some problems inside the context. But let's imagine that you have them, so you just fuse them together to create a proxy of the genome, and you did a Hi-C experiment and you created a map, just like what you would in the normal situation. So here's an example of the map that you could get this way, right? So here are these 3 contigs, the 1, 2, 3, and you can see the, okay, I'm going to do it this way because I don't see the, oh yeah, here. Yeah, this works also. So, right, the 3 contigs that are supposed to be contiguous sequences, you did the experiment, you did the contact map, but this map doesn't quite look like the human contact map we saw a few slides up. Right, what's the problem? Let's try to explore. Right, so one thing that you can see here is there is this weird pattern in the third contact where there's a bunch of things. There's a big square upstream and a big square downstream, and then these two bits, they don't seem to be talking to each other at all. So what's going on, right? So it's possible to try to like think of weird ways in which the polymer could do that, or we've never seen the polymers, any biological polymers do anything like that in many years of doing high-C experiments. So the reasonable assumption there is that it's actually not the polymer, it's actually a mistake in the original contacting, right? And what we need to do is we need to cut in order to get to truly contiguous sequences. All right, okay, what else seems weird? Another weird thing is that there is this off-diagonal signal here and here, and of course this map is symmetrical, right? That basically suggests you that this bit that we just cut out, it has a lot of 3D contacts with the stuff upstream here. And again, right, that probably means that if you just move it closer in 1D, right, that off-diagonal signal will become immediately near the diagonal, and this map looks much more like what you had to do. So you just like fixed a translocation type problem. So this map looks much more like the human map that you were staring at a few slides up, but still not quite. So notice this, like there's this bow tie-ish motif here, right? So it suggests that the beginning of this square bit that we just moved, it talks a lot to this stuff immediately downstream, and the and talks to the stuff immediately upstream. Any ideas? What might be going on? Yeah, I can see a few people doing the right thing with their hand. That's right. Okay, so right, if we would have just flipped, right, this immediately, the stuff that had a lot of contacts become close in 1D proximity, so the 3D the 3D frequency becomes, behaves well with the 1D distance, and we get something that is more or less looks just like the human map that we saw before, and for good reason, because you guys have just assembled 2 human chromosomes. So, humans are really good at noticing these kinds of patterns, these kinds of mismatches, these kinds of gradients, and fixing them as well. So as part of, our effort, we basically created a tool that allows one to do this sort of thing we just did step-by-step interactively. And would be good to have that a little bit louder, but yeah, because so as you do this, it helps to have fun music in the background. Apparently, yeah, I was considering to like incorporating the kazoo, but then I wouldn't be able to really comment on what's going on. So yeah, if you can come up and add some music, like clearly, you would have been more reliable. So what are we doing here is that, right, we're doing exactly what I showed you with the 2 chromosomes. It's just a little bit sped up. So what I loaded here is the contact is a draft contact map for the Medicago clover. It's the same kind of map, the same kind of, randomly arranged contacts that, you had a map that didn't look quite what you expected from a normal high-C contact map, and then I went in to edit misjoins, move things together in 1D, things that seem to have a lot of contacts, invert, bits within the individual chromosomes, and then it's just, if you were keeping track of that time over there, that was about 10 minutes, and 10 minutes in which you went from here, this stuff on the left, to this stuff on the right, like a bit of a misordered mess of stuff, to like 8 beautiful chromosomes that are much easier to interpret. And of course, even with a lot of fun music, you wouldn't want to do that if you have hundreds of thousands of pieces, so that doesn't, neither the kazoo nor the Tetris music saves that sort of situation. So for those purposes, we've taught the computers to do more or less the same sort of thing. So here what you're looking at is the Aedes aegypti draft, the kind of, like the one that was presented on the front page of the New York Times, just a little bit older, before that time, that we do the same thing and put together into 3 long end-to-end chromosomes for the yellow fever mosquito. It's the same high-C data, right, the before and after. And, just to give you like a sense of scale, this experiment has cost us maybe about $200. I don't know if this was, the cheapest science paper ever, but it certainly was the cheapest science paper coming out from our lab. So the whole scale of like how cheap you could do these experiments and what you could do with this sort of data really led us to feel like it was time to start doing this at scale, to start doing, comparative work at scale. So we've kind of rallied some support from the zoos in Houston and in Texas more broadly and outside. So to build a sample acquisition network, we were lucky to gain some support from Illumina and like some computational companies in robotics, and collected a huge collection, of more than 1,000 species of cultivars, as part of this DNA Zoo Consortium, which is the consortium among zoos, academic institutions, individual researchers to rapidly generate and release chromosomal genome assemblies based on Hi-C. And so, we assemble a lot of genomes this way. So at this point, we've shared about 30 genome assemblies. We try to do this more or less every week. All data and methods, importantly, on open source, so everybody can go and explore this data. And you've heard already, like from Eris, for example, about the types of studies that are being done on this data, including, like we're teaching the pigs fly. But what I'm going to talk to you about is another study that we're trying to do with this kind of like large comparative data we're creating as part of the DNA Zoo Consortium. And a study that we've recently published with colleagues from the Netherlands and the Center for Theoretical Biological Physics. And so in this particular study, we're curious to understand how the species fold, specifically the eukaryotes, how do the different species fold across the tree of life. And remember that as we assemble these genomes, we assemble them with the Hi-C, but that means that then, once we're done with the assembly, we can re-examine the Hi-C to understand how any particular genome folds in 3D, what is there interesting, and if there's anything we can learn about it. So in order to try to figure something out about the evolution of eukaryotic nuclear architecture across the tree of life, in the spirit of DNA Zoo, we rallied, some samples from our zoo collaborators, and we've put together the contact maps from 24 different species that include plants, fungi, a bunch of animals, including 7 out of 9 a major animal phyla, a bunch of chordates, and overall basically a pretty dense sampling of the eukaryotic tree. And we're lucky 6 species among those 24 were in a pretty good shape that we could use them sort of offhand. The rest we had to assemble either from scratch or upgrade from the existing drafts. And so what did we learn once we assembled and mapped these all of these species? So what we saw is kind of an embarrassment of riches. You take the zoo in, zoo out sort of thing, because what we saw is a whole collection of very different maps that are very difficult to interpret because they had different chromosome sizes. Some of them had a lot, some of them had little, some of them had a lot of interchromosomal, some of them had little. It was just very, very difficult to interpret. It was clear that we're not going to get anywhere until we develop some kind of a common approach to try to compare. So in attempt to do that, we've developed something that we call the aggregate chromosome analysis. Basically, the idea is that we, average the maps in order to create a contact map that shows how 2 typical chromosomes in a species form. So the typical chromosomes being the chromosomes in which the centromere is right in the middle, the chromosomes are of the same length, and sort of that takes you from this map on the left, that of 3 chromosomes of different type and different length, and the centromeres are in different positions, to something like this, where you have these 2 chromosomes with the centromeres right in the middle. Okay, so once we did this, what will we start? Oh, sorry, I forgot to say that. Of course, those chromosomes are symmetrical, and you can kind of remove half of it without the loss of any information and move to this triangular representation that is also very popular in papers on contact mapping. So, if we apply this sort of approach to the contact maps that we saw from across the different eukaryotes, you'll start finally seeing something that seems to be recurring. So the recurring features that we start seeing are shown here. So in some maps, we saw that the maps look something like this, where it's there was this bright dot in the middle, like in the ACA map that I showed you. And that, of course, means that the centromeres talk to each other a lot. Right, so this suggests something like the following arrangement. In some other contact maps, there were these peaks over here. That suggests that there were telomeres talking to each other a lot. And some contact maps had this bright line over here that suggests that there was some kind of a centromere to telomere axis. And in some contact maps, there is, of course, nothing at all in the middle and just 3 triangles, which basically suggests that the chromosomes occupy their individual subvolumes and they don't seem to intermix all that much. All right, so, just give these triangles a little bit of a squeeze, and let's try to just to kind of neatly arrange them around the phylogeny tree, and this is what you will get. So, right, so what you're looking at here are these, ACA contact maps for individual species that are shown here. Here's one for the Tamar wallaby, one for the chicken, one for the Burmese python, et cetera, et cetera, et cetera, et cetera. And you go like this for the last 2 billion years. And so staring at this map, we learned, I guess, 2 interesting things. So one thing that we learned is that, these features that we saw, they don't seem to be behaving like your typical good phylogenetic characters. So pay attention, for example, to the centromeres over here, because they're really easy to see here. They are in the mushrooms, here they are in some basal animals, here they are in the dipterans over here, and here it suddenly emerges again in the African clawed frog. So not like what you'd imagine, it appears in some sort of phylogenetic branch and just continues doing something there. So that's one thing. Another thing that we learned is that these features that I showed you, they co-occur. So pay attention, for example, to this mosquito map, right? Here's the centromere peak, and there are these lines over here that correspond to centromere to telomere axis. And if you examine like how exactly do these features co-occur, what you'll see is that the first 3, the centromere, telomere, and telomere to centromere axis, they tend to co-occur a lot. While if you have chromosome territories, you usually don't see any of the other 2. So what we really should be talking to based on this is not the 4 different features, but rather actually the 2 different types. The one type is in microscopy usually referred to as like rabble-like architecture, and the other one is the chromosome territory type architecture. And of course, what it seems that you're having across the tree of life is just switching between the one and the other. It's not entirely clear what's going on. Of course, we were interested to understand the mechanism by which this switch is happening. And so in order to try to look for potential culprits, what we thought towards is that, well, the rabble kind of looks a little bit like mitosis, because, right, if you think what happens during mitosis when the cells divide, the chromosomes gets dragged by the centromeres, which, naturally creates this clustering of the centromeres, clustering of the telomeres, and the whole telomere to centromere axis. So we started systematically looking through different machinery that handles mitosis to try to see if we see any correlation between the mitotic machinery and the our type 1 or type 2 architecture. And we did find some. So the thing that we found is that it seems that the type 1 correlates basically with the state of condensin 2, which is this multi-unit protein that condenses the mitotic chromosomes by doing loop extrusion, does the lengthwise compaction. So what you're seeing, back to this mandala figure of ours, what is added here are these black dots over here that represent 5 subunits of condensin 2. And anytime that you see a white dot, it means that one of these subunits is broken. And what we noticed is that very often the type 1 architecture, the rabble one, would have some of the condensin broken. In fact, anytime that you had the condensin 2 broken, you would have the rabble. And so clearly we had some kind of a correlation, but did we have causation? Of course, the ultimate test for causation is a forward experiment. So in order, right, what you'd want to really do is take some species that is type 2, that is chromosome territories, break condensin 2, and see if that turns it into rabble. So in order to attempt that, we turned to our colleagues at the Netherlands Cancer Institute, and they had this wonderful system for, the Dagger-induced rapid removal of condensin 2. And, Claire, shown here, did the experiment. We were super excited. In the first experiment, showed us nothing. So what you're looking at here is that, all right, so she measured the number of centromeric foci per cell, basically with the idea that as they start condensing, the number should go down. And nothing went down, so we were, listened to some sad songs, contemplated alternative careers, and, all that. Until, we were trying to think, what may be going wrong in this case, and we thought that, well, okay, we're depleting in the interface. So what we maybe should be doing is we should let these cells go through the mitosis. And indeed, once we did that, this is when we started seeing the clustering. So we're incredibly excited. We've confirmed that this experiment works. With Hi-C, here's the ACA, the aggregate view. You can clearly see the increase of this centromere to centromere signal, the disruption of chromosome territories. So you basically took, right, a species, a human, at least in some respect, you basically converted a human to a fly. For the Jeff Goldblum fans out there, if there are any still left, the Brundlefly would approve. But we did, a bunch more stuff to, of course, describe what's going on in detail in these new cells, but it seems that more or less nothing really kind of got majorly disrupted. The local architecture was not much affected. The gene expression was not all that much affected. But interestingly, so what the microscopy showed, what was going on is that the centromeres were clustering at the nucleoli, but it showed that the heterochromatic domains were merging. And that was sort of what has led, has served as a guidance for the model that our colleagues at the Center for Theoretical Biological Physics have developed to try to describe how exactly kind of the lengthwise compaction can lead to, who knew, the arable architecture disruption. So the model has been, the development has been led by Sumitaba, showed here, and I understand that Michele Di Piero will talk in more detail about this model on Thursday, but very, very simplifying, right? Like you kind of think of this, that as you get a lot of lengthwise compaction, the heterochromatic sticky heterochromatin and the centromeres, it gets shielded, it can't merge, as you go through mitosis, and then as a result you get chromosome territory as well. If you have like long floppy chromosomes, the heterochromatic domain gets exposed, it's sticky, it sticks to the rest of the stuff, and you get the rabble. And, the simulations showed that the model is consistent with the experiment. Here's the results of the simulations again in this like ACA framework. And again, which more or less shows again and again that if you have short and rigid chromosomes, they form chromosome territories. If you have long and floppy chromosomes, they show the rubble. And of course, the standard, a question you should expect from reviewer 2, is there any other ways to get to long floppy chromosomes? So lucky us, the nature did the experiment. We didn't have to come up with anything else. So, right, the beautiful experiment that the nature did is, it created the long floppy chromosomes by the means of fusion. So there exist these two muntjacs, one the Chinese muntjac and the Indian muntjac, and one has 23 chromosomes, which is a pretty typical karyotype for a mammal, and another one has just 3 chromosomes, so it's the smallest number among mammals. And the way that it happened is that it happens through multiple fusions, but they're really close in the sense that the content is the same. They're close enough that you can hybridize the two. But yet, right, as you can see, the one with the short rigid chromosome, it has chromosome territories, while the other one shows clear signs of rabble. So we're very excited. I know that I'm out of time, so I'm not going to spend time on the conclusions or just more details in the paper, but instead I'm just going to say that this is an example of the exciting stuff that you can start doing when you have a lot of chromosomes assembled and a lot of 3D information for the species, so that you can start doing comparative genomics, not just in 1D, but in 3D. And if you have ideas and if you're interested, the data is out there, and of course we'd be happy to collaborate. So please reach out if you're interested in comparative 3D. And thanks to all the zoos and all the partners, and of course thanks to all the collaborating labs, because this was really, a really powerful 3-way collaboration with a comparative, the forward experiments, and the modeling. Thanks so much for your attention.
Thank you, Olga, for a beautiful talk. We have time for a couple questions. Let's start.
Thanks a lot. It was a nice talk. So actually, I was wondering how many species in your zoo were holocentric, if any?
We didn't do any specific counting, but there were, I think, 2 in this particular paper. But so we do have some Lepidoptera, and we do have, the C. elegans, so that the standard ones. We do have, I'm not sure if we have a whole lot of plants, but so basically we do have, but we haven't had a project that has purposefully said we're going to go for holocentric versus monocentric, but of course it's super interesting. Yeah.
So you don't see any specific feature associated with holocentric? I mean, apart from the fact that they don't have a central.
All the holocentric turned out as type 2, so basically chromosome territories. That I can tell you. So it's just, not entirely surprising, I guess, if we do believe that rabble has something to do with the mitosis, right, and the way that they're dragged. But, so far we have not found an example where, contrary to our thinking in this space, that the holocentric species would form anything that looks like a rabble.
So the thinking that the rabble configuration, for example, if you take the Drosophila, yes, there is rabble configuration, but there's also nuclei in different cell types that don't. So I guess that the tissue that you choose to do your high-C analysis may, to some extent, determine the kind of configuration that you find. So have you, I mean, for some of the species, tried different tissues, different stages, different cycling speeds to see if this makes any difference?
Yeah, that's a very good question. Of course, across such different species, we weren't able to really control for that, but we thought that having a forward experiment where in exactly the same cell type, right, like you can convert one for the other, sort of compensated to a degree for that. We suspect that it's basically the matter of, you know, as you have some cells, if you have basically like very fast cycling through like the cell cycle, you will have a more pronounced, or when you have cells that sit there, the structures tend to diffuse, and so probably in like highly differentiated, long-living tissues, you'll have a little bit more of that. But I suspect that still, usually again, it's basically more pronounced or less pronounced, but not necessarily that like it's really a completely different architecture type.
I just have one short question, because I think the idea of using high-C in order to do get the sequence of genome, how good is that sufficiently, or do you need still some long pairs in the end, or that's sufficient to go all the way to the end?
You know, in general, to keep everybody happy, usually the answer is that, you know, the more the merrier. So basically, right, it's not like you're losing anything by introducing additional data types, you know, even if for validation. But we have shown that if one does want to use resources for something other than assembling genomes, usually you can get about with just the cheapest data type there is, you know, for mammals, for example, just let's say like 300 million short paired end short insert reads and 100 million reads of high-C, which I think right now costs like about $70 or something. You can get a chromosome length mammal.
Okay, that's a good story, and I think it's a beautiful story. Thank Olga, and I want to thank all the other speakers on the session, and it was a pleasure to chair. Three great talks.